is a professor emeritus of Trinity University, and he is my go-to guy for explanations of everything scientific, particularly now when we're also concerned with the virus. It's all about biochemistry. So I asked him today when we were talking to explain about the test. There is so much controversy over who gets the test and how it's done, and he talked about the procedure and helped me understand a little bit more about why this is such a critical part of what we're going through. Bill, thanks.
Sure. So what happens when you go and get a test is they take a swab and they put it through your nose into the back of your throat, and you will get some, if you have the virus, you will get some virus particles on the swab. They'll then put that into a solution of chemicals. And the idea of it is simply that this solution, this group of chemicals, is going to cause the virus particles to fall apart into its separate components. And so in the virus you have what's called an RNA molecule, a type of nucleic acid, and then you have proteins, and you have some fat molecules and other kinds of molecules, and this solution just causes the virus particle to come apart, and then you take this and take this solution of all these different components, spin it down in a centrifugal field, and it allows these components, which have different sizes and shapes and weights and so forth, densities, to separate into different layers. So you can get out the RNA, but it's not much RNA. It's the RNA that they're after to identify the virus. And so now let's assume, just for simplification purposes, that you have one molecule of RNA that you've extracted from the virus. In order to really know that this is viral RNA, you have to make a lot more copies of this in order to be able to test for it. So what they use is an amplification reaction that's called a polymerase chain reaction, and you just mix that strand of RNA with some other chemicals that are designed actually to copy a certain part of the one or more of the genes in the viral RNA. So you start with one strand, and then you put this into these chemicals and enzymes, and it makes a new strand, which actually is DNA. It has the same sequence as the RNA, but it's a strand of DNA. And then you separate those two strands, and then you make copies of each of those two. And so if you do this about 40 or 50 times, you will end up with millions of copies of the original nucleic acid strands. Some will be RNA, some will be DNA, but they all have the gene of the virus located in them. So you've made millions of copies, which you can now test. You put in a fluorescent molecule that binds to the DNA, and it lights up in the machine, and based upon that fluorescence intensity, you can verify that you have the viral nucleic acid present. And that's pretty much all there is to it. Now that takes, in the way it's been done in the past, that can take several hours to do. By the time the sample gets to a lab somewhere, which might take a day or two, and then it will get treated or handled by the staff there. It might take another day. It might be 4 or 5 days before you're going to get results. But there is new equipment that is being produced by one or more companies in the country now that can be used on site and reduce the time for making the millions of copies of the DNA to less than an hour. And so that's what everybody has been hoping would become available we've heard that should be available very soon, and hopefully they'll be able to do a lot of testing of those people who are at the front lines fighting the virus, especially in the hospitals. So that's what it's all about.
What is very soon? Next week? Next month?
Well, it was supposed to be available sometime at the end of this week. I haven't heard whether those rapid tests are available or not.
We can all hope that they are, but when they come available, we'll know how they work. Thanks so much, Bill. Thanks for your explanation.
You're welcome.